to the NPTEL lecture series on animal physiology. So, we are in section 12. So, today we will be starting with the lecture 4 of section 12, where we are in the gastrointestinal physiology, and we will be talking about intestine and pancreas. If possible, we will do the liver. So, as of now we talked about, first of all let us have a brief recap, 2-3 minutes. So, initially I give you the whole outline of from mouth to anus, how the food is passing through. So, initially in the mouth we chew the food using our teeth, then there are taste buds along the tongue, and it is out there the salivary gland plays its role. It secretes a series of enzymes which helps in softening the food, and then through peristaltic movement the food travels down to the stomach. In the stomach it is exposed to an extreme acidic environment, and in that acidic environment series of chemical reactions happen. The protein gets denatured, they got broken down, and then food is further crumbled down into very smaller pieces. From there the bolus or the whole lump of food moves to the small intestine. It is in the small intestine where the maximum absorption takes place. So, while we did about the cross section of the stomach, we saw how the brush border like structures were there. A very similar kind of structures indeed exist in the intestine, but much more higher surface area, and the cell types which involved in it are different. So, what we will do essentially the same way we did for stomach, I will draw the whole anatomy, and from there we will talk about the cross sections, and we will talk about the different cellular structures which helps in the absorption process. Okay. So, getting back another slides. So, we are into section 12. So, we will be talking about now intestine, and another thing what I am going to talk about today will be the pancreas. So, if you people remember, while we talked about endocrine system, I told you that there are series of hormones which are secreted by the pancreas, and we will discuss those in the digestive system or in the gastrointestinal physiology. So, I think now since we are doing the gastrointestinal physiology, once I finish the intestine, we will move on to the pancreas. Okay. So, these are the two stuff what we are going to deal today. Okay. To start off with small intestine. Okay. The anatomy of small intestine, if you look at it. So, the food from the stomach moves, and then there is a series of structures like this. It's a long loop like structure which is kind of something like this. Okay. So, this is a huge loop structure, and of course, then it leads to the large intestine, which kind of is fairly bigger diameter, which is surrounding it, which is also called the colon, before the food is kind of excreted out through the anus. And here you have the colon, and this zone what you see, this whole tubular structure, this is almost 20 feet long, or which is almost in terms of 6 meters. Okay. And the diameter range from 4 centimeter to 2.5 centimeter. So, these are some of the logistics, and this whole small intestine is divided into several parts. So, if I redraw this, it is something, it looks like so you have the entry port from where it is entering closest to the stomach, that is called duodenum. Then you have next part which is called jejunum. Then you have sorry, just bear with me. This is, I just made a mistake, and so okay. Let me redraw it, then it will make more sense. So, from here the food is entering from the stomach, and now it is traveling all along the small intestine out here. Now, it is moving on to the large intestine out here. From here starts the colon. Okay. So, the first part is called duodenum. Then you have the jejunum, then you have ileum, and then you have ileocecal valve. This is the overall anatomy. If I have to say how the intestine is divided, intestine is divided into 4 parts: duodenum, jejunum, ileum, ileocecal sphincter or ileocecal valve, from where the food moves on to the colon. So, it is a huge tube, and this whole tube, if I had to kind of give an analogy of the histology, the whole tube has borders like this all over inside, something like this, hairy border all over, and this border is essentially what is termed as the brush border, and those projections like a structure what you see like this, which are projecting out, those projecting structure are called villi, villi, villi. So, if you look at this, so essentially what is happening, so suppose a food comes, the food particles are moving like this all along the villi. So, you are enhancing the surface area many folds by creating that villi like structure, and this is how the biology designs its structures. So, that in a very limited space you can maximize your output. So, coming back now, what we will do after giving you this anatomical features, I will move on to give you an idea about the histological aspect of it. So, we will take a cross section of this and we will talk about the histological parameters. Okay. So, coming back to the slides now. So, this is how it looks like. If you look at the cross section of one of this is how it looks like, and what you will see essentially lot of, and if I could draw it in 3 dimension, it is something like, so you will see all over the place there are brushes, just like your toothbrush you see, just like a toothbrush you will see lot of these structures, and these structures are called villi, which is the brush border, and this whole raised structure is called plica, P-L-I-C-A. So, what essentially is plica? If I going by the definition, the plica is basically the intestinal lining. Basically, the technical definition of plica is intestinal lining bears a series of transverse fold called plicae. Transverse fold called plicae. So, it is very similar to the structure what we have explained in stomach, which is called rugae. If you remember that structure in the intestine, I was showing you in one of the slides that surface is called rugae. It's this, and in the case of intestine, is called. The structures are almost same, but it's just they are functionally different from each other. Okay, because at this stage, the whole idea is to absorb as much nutrient as possible. That is the major. So, automatically when you have to absorb, so you can understand if you look at the picture. So, this whole thing will have lot of blood vessels all around it. Likewise, these blood vessels are continuously absorbing whatsoever nutrients which one is kind of going through the intestine. So, this is continuously getting absorbed from the blood vessels, and it has to be transported for synthesis of different kind of molecules. Okay. Now, coming back to the slides, let me move on to the next slide. So, now what I will do, I will draw the detailed structure of, I showed you this. Now, what I will do, I will just take one cross section out here from here, one section, and in the next slide I will show you how that looks like. So, something like this. Okay, let me just okay, fine. Okay, so this is the bottommost layer, which is, and underneath also there is a layer which is called serosal layer. Okay, this black what I am. One second. Okay. This base layer is called serosa. On top of serosa, you have muscularis externa, and muscularis externa is something like this. This layer is called muscularis externa. Next layer. On top of that, you have the layer called submucosa. So, here I am into the submucosal layer, okay, which is. So, how about we give different colors to them, so that you can identify. So, okay, now we are entering into the submucosal layer. So, submucosal layer has the blood vessel all over the place. This is submucosa. Okay. On top of submucosa, you have something called a layer, very thin layer, which is called submucularis submucosa. Muscularis mucosae. Muscularis mucosa. Okay. And on top of this, you have the villi like structure. Likewise. So, this is the total cross section of it, and underneath you have something called lymphoid nodules, and you have out here the intestinal gland, or this is also called crypts of Lieberkranz, intestinal gland. This is also called crypts of Lieberkranz. There are two names for that, this base region. And now what we will do, and these are the individual villi. Okay. Now what we will do, after giving you the outline of this structure of the cross section of the intestine, what I will do, I will give you the individual villi, how the cross section of the individual villi looks like, and where are the blood vessels, how the blood vessels are pulling the nutrients from individual villi, because these individual villi. So, what is essentially happening, if you look at it, this. So, the food is, let us represent food with yellow. So, the food particles are all over the place, this is moving, the food particles are traveling all along like this, and at these zones all the absorption is taking place, all the different kind of all the nutrients which has to be absorbed, they are getting absorbed here likewise. And once they are absorbed, they are further transmitted through the blood vessels which are present there. So, now after this we move on to the next slide, where I will draw the structure of the individual villi and we will highlight all the different structural features into it. Okay. Let's move on to the next slide. Okay. So, now let us look at the cellular structure of. So, the overall structure is, one second, overall structure is clear to you people. Now, let me okay. So, this is what you see, what I am drawing now is the lymph vessels which runs parallel to the blood vessels. You saw the lymph vessel which I was drawing. So, these are the lymph vessel, and now this is all surrounded by the blood vessels. So, they come parallel to them. Okay. So, those blood vessels are out here. Like this, they are all surrounding them, and in continuation you have the venules which are the veins which are in blue, they are running in parallel, and that's it is it. Okay. So, the red ones, so these are the veins, and here you have the arteries. Okay. Now, is the cellular layer, which is use a different kind of color for that. Okay. Okay. The cellular layer, you could see, could be I'm just using a slightly lighter color out here, so that I can. So, this is how the cellular layer is, and on top of that green what you are seeing the lining, that is where started all the brush borders. Now, what I am drawing now is just like your toothbrush, if you look at it. Okay, this is how a brush border structure looks like. Okay. So, the cells, what are the different terminologies for the different cells out here? Some of these cells which are at the top, what you see, these are called goblet cells. Then you have the epithelial lining. So, most of these are epithelial cells. Then you have this part of the lymph vessels is called lacteal vessel. Lacteal network. This is the capillary network, and this part is called lamina propria. But in this process, you must have observed that I have not highlighted one thing yet, that is the nerves, because it is innervating, it is innervated very thoroughly by the nerve vessel. So, where the nerves are present. So, now let me introduce the nerves, they are all over this. So, these nerves are moving all along like this. These are the nerves, all the sympathetic, parasympathetic, what you see. So, these are the nerve endings. So, this is how complex individual villi are, and there are thousands and thousands and thousands of villi all along the small intestine, and the food particle just like this, if I have to use a thick this food particle is moving like this out here on these surfaces. these are the food particles. So, they are following this route, moving like this, each one of them. And in that process, what is happening essentially is all the material what has to be absorbed are getting absorbed here, and they are being taken up by the blood vessels, and they are being taken up by the blood vessels and used for energy production. So, this is how the complete profile of the intestine looks like, and this is how the intestine is absorbing all the different kind of material along its surface. Okay. So, now let us get back to the slide and try to tell you some of the different intestinal secretions. What are the different intestinal secretions which are taking place? And so, roughly 1.8 liters of watery intestinal juice enters intestinal lumen per day. Okay. And much of this being used for breaking up the for promoting the movement of the food and all other things. Okay. And the intestinal movements includes, there are two sets of movements which are involved in the intestinal movement. One is called gastroenteric reflex, which is regulated by the nerves reflex, and I have talked about the reflex circuits already to you. And there is something called gastroileal reflex. Okay, so these are the major intestinal movement which is taking place in the intestine. Okay. So, this is the overall understanding of intestine, what I expect you people to get. From here, what I will do, I will move on to the structure of the pancreas, what I told you. Okay. I want to say. So, again with the pancreas, we will do the same thing. We will talk about the first of all the position of the pancreas, where exactly it is located, and we will talk about the secretion made by the pancreas and the role played by pancreas in the process of digestion. Okay. Let us get back to the slides. Talking about the pancreas, this is the stomach, from the stomach out here. Okay. And here start duodenum. So, this is stomach, and here starts the duodenum, or the part of the intestine, small intestine. So, the pancreas lies somewhere here. So, okay. So, organ like this constitute the pancreas. So, the structure, the way I have drawn it is fairly similar to that. It's kind of if you take pepper and you cut it. So, the top part, it is almost like that, it is kind of inside a groove. Okay. So, what the pancreas does, let us come back to the slides. Okay. So, if you look at the pancreas, so this is very well innervated by the blood vessels which are coming all over, and mind it, this is also have a lot of endocrine function it has to take care of. So, this is the body of the pancreas. Then you have the pancreatic arteries, these are called pancreatic arteries, and out here are the, if I had to do the cross section, are the exocrine cells in pancreas, which are also called acines, and those acines are something similar to, let me draw the acini in the next slide. Acines are very interesting structures. So, these are structures like, so there are vessels all along likewise, and within those vessels you have circular structure like this. Let me draw it and it will make more sense to you people, like this. Okay. And these are further okay. These further have the cells which are arranged like this. They have this duct which are attaching. So, all of them have something like this. I will come to the labeling and everything. So, these are the ducts and these are the exocrine cells in pancreatic islets. Okay. So, out here there are some series of cells which are more like this. They have their own vessel and so these cells which are present. So, these are the ducts what I have already drawn, okay. And you have the connective tissues in between which all over the place. These are the connective tissues. And you have the so these are the exocrine cells in the pancreatic acini. These are the exocrine cells in pancreatic acini, and you have the endocrine cells which are these ones, which are the endocrine cells in pancreatic islets, which are also called islets of Langerhans cells. So, these are the cells, this one which I have drawn in yellow or slightly muddish yellow are the ones which are called islets of Langerhans or pancreatic cells, which are involved in the secretion of a whole series of different factors which help in the digestion. So, what all pancreas is secreting? So, one is, so P stand for pancreatic, okay. Alpha amylase or carbohydrates, basically this is breaking down the carbohydrates. Then you have pancreatic lipase, which is essentially breaking down the lipids. Then you have nucleases, which is essentially breaking down the nucleic acid. Then you have proteolytic enzymes, which is taking care of all the proteins and peptides, breaking all the proteins and peptides. So, if you look at the pancreas, I have told you, I have given you at least 4 different enzymes which is taking care of the fats, lipids, carbohydrates, proteins and the nucleic acid. Apart from it also, it has the endocrine function. So, now what we will do, we will enlist or enumerate all the endocrine secretion which are made by the pancreas. Okay. So, that includes this. So, these are all the proteolytic enzymes we have talked about. Okay. So, now we will talk about a few other enzymes before we get into that. So, one is trypsinogen, chymotrypsin, procarboxypeptidase, proelastase, and these are the series of additional enzymes which are secreted among the under the major class of the one which will be breaking down all the different proteins. Okay. So, after this we are left with few other aspects which pancreas is going to secrete, which helps in the digestion of the food. Okay. So, let us just go through what all we have covered. So, we talked about the intestine, and in the intestine we talked about the structure of the villi, where all the absorption is taking place. Then we talked about the anatomical location of the pancreas, and we talked about the major secretion made by the pancreas and the histological structure of the pancreas. So, what we will do in our next class, we will talk a little bit more about the pancreas, about its endocrine roles, and then we will move on to the liver, and we will finish up with the liver, and then we will move on to the large intestine. Okay. Thanks a lot.